We often hear about these exciting new medical treatments, and it's easy to just assume that once a discovery is made, getting it to patients is simple.
Right, like it's a straight shot from the lab bench to the pharmacy shelf.
Exactly. But have you ever really stopped to think about all the hidden stuff, the complexities, especially with something like peptide therapeutics?
Absolutely. And that's really what we're digging into today. We want to explore those significant hurdles involved in actually making these promising peptide therapies.
Not just discovering them, but manufacturing them at scale.
Yeah, at the scale needed to actually help lots of people. We'll be looking at, how they're synthesized, the molecules themselves,
Okay.
and also the really critical need for strict quality standards. It's called cGMP,
TGMP, right? Good manufacturing practice.
current good manufacturing practice, yeah. And then there's the whole regulatory validation side for these active ingredients, the APIs, the actual drug part.
Got it. And we've looked through quite a bit of material for this scientific articles, pharma development guides, regulatory stuff.
We have. The idea is really to give you, our listener, a clearer picture of these often unseen challenges, bringing these innovations from the lab to the patient.
Okay, so let's jump right in. Maybe start with the basics. What exactly are peptide therapeutics? What makes them interesting?
So peptides, they're basically short chains of amino acids. Think of them like, well, smaller cousins of proteins in our body.
Okay, smaller than proteins.
Yeah. And what's cool about them is they can sort of fit into this unique niche in medicine. You can design them to be super specific.
Target very particular things in the body.
Exactly. Target specific biological processes, which often means they can be really potent, but potentially with fewer broad side effects compared to traditional small molecule drugs.
Interesting.
But that complex structure, that's also what makes manufacturing them pretty challenging.
Right. So they hold a lot of promise, but that promise comes with some difficulties in production. Where do things start to get tricky, typically?
Well, one of the first big roadblocks is just the synthesis, actually building the peptide molecules.
Making the chain.
Yeah, it sounds like maybe just linking amino acids together, but doing that correctly and on the kind of scale you need for medicine. It's way more involved than making, say, a lot of standard small molecule drugs.
Why is it so much more complicated? What's the specific challenge there?
Well, imagine you're building a very specific, very long necklace with different colored beads in a precise order.
Okay.
For longer peptides, making sure every single link is right, and you end up with the exact sequence that gets harder and harder.
Any mistake is a problem.
A big problem. Any mistakes in that process can lead to impurities.
Impurities.
Yeah, molecules that have the wrong sequence, or maybe they're missing bits, or they have extra chemical groups stuck on them. And these impurities can really mess with the drug's safety and how well it works.
That makes total sense. Like misspelling a word in a long sentence, it could change the whole meaning or just be nonsense.
Precisely. Now, the most common way people make these peptides, synthesis-wise, is something called solid-phase peptide synthesis, SPPS.
Okay.
Yeah. Now, our sources don't dive into all the nitty-gritty chemistry details, but the basic idea is you build the peptide chain step by step while it's attached to a solid support.
Like attached to little beads or something.
Exactly, tiny beads in the reaction vessel. And that attachment makes it easier to wash away all the leftover chemicals and byproducts after each step. You add an amino acid, wash, add the next, wash.
So you're kind of growing the peptide molecule on these little anchors.
That's a good way to put it.
Yeah.
And while SPPS is, super common, taking it from a small lab flask to producing kilograms, maybe even tons of a peptide, that's where the real scaling challenges come in.
Right. What happens then?
Well, for instance, making sure each step is really efficient, like nearly 100% complete across a huge batch. That gets much tougher.
Consistency is harder.
Much harder. And the quality of the solid support itself, those beads, can become an issue at large scale. Plus, the sheer amount of chemicals you need. That becomes a huge logistical and cost factor.
And are there other ways besides SPPS?
Oh yeah, there are other methods, like liquid phase synthesis where everything's dissolved. But SPPS is often favored because cleaning up the product, the purification, can be relatively simpler, even with its own scale-up headaches.
I feel like I've also heard about microwaves being used sometimes. Does that help with these scaling problems?
That's a good point. Microwave assistance. It is a technique used in peptide synthesis.
Yeah.
They can potentially speed things up, the chemical reactions, maybe improve the yields.
Okay.
But actually applying that effectively at a really large industrial scale, that still has engineering hurdles. Getting even microwave heating throughout a massive reactor isn't trivial.
So it's promising, maybe, but not like a magic bullet for large-scale production yet.
Not universally, no. So
Yeah.
bottom line, whichever synthesis technique you use, getting that high purity and a good yield when you're making huge amounts, that remains a major challenge.
And you mentioned purity being critical because of impurities affecting safety and efficacy.
Exactly. Any unwanted stuff in that final drug product could, best case, make it less effective, worst case, cause unexpected side effects or even be toxic.
Which is obviously something you absolutely have to avoid.
Right. And that leads directly into the next big challenge, actually scaling up the entire manufacturing process, not just the synthesis, while keeping that quality control airtight.
Taking it from the lab process to a reliable factory process.
Yeah, translating something that works beautifully in a little glass flask to something that works reliably, consistently, day in, day out in a giant stainless steel vessel. That's a massive undertaking.
What kind of specific problems pop up during that jump, lab to factory?
Oh, several things. First, just getting your raw materials. You need large volumes, consistently high quality. Sourcing that reliably can be tough.
Supply chain issues.
Big time. Then, the equipment itself. Industrial reactors behave differently than lab glassware. Heat transfer is different, mixing is different. So you often have to significantly re-optimize the process.
Adjusting all the parameters.
Reaction times, temperatures, how you purify it.
Yeah.
Everything might need tweaking and revalidation when you're dealing with much bigger batches. You need a process that's robust,
Robust meaning.
meaning it can handle slight variations in conditions or materials without the final quality suffering. It has to produce the exact same quality product, meeting specifications batch after batch after batch.
It's not just about making more, it's making more perfectly every time.
That's the goal. It sounds like a really delicate balancing act.
It absolutely is. And this is where that term cGMP becomes so important. You mentioned it earlier. What exactly is cGMP and why is it so central here?
Right, cGMP, current good manufacturing practice. These are basically the rules, the regulations, like in the US, you have 21 CFR parts 210 and 211 that set the minimum standards for making pharmaceuticals.
Minimum standards for everything involved?
Pretty much. They provide a framework to ensure the drug's quality, safety, and effectiveness. It covers everything from the building itself, the equipment, how personnel are trained, the documentation, all of it.
So it's like a comprehensive rule book for safe and consistent drug manufacturing.
That's a good way to think about it. And for peptide APIs, some cGMP aspects are especially critical. Let's talk quality control, for example.
Okay. Testing things.
Rigorous testing at every single stage. You test the starting materials, you test things during the synthesis, the intermediates, and you test the final API. All to make sure they meet preset quality standards.
Specifications.
Exactly, specifications. And there are international guidelines, like ICH Q6A, that help define how you set those specs. You need really sophisticated tests, analytical methods that can precisely identify the peptide and measure any tiny amounts of impurities.
Impurities, again, they seem to be a constant theme here.
They really are a primary focus in peptide manufacturing because the synthesis itself can create all sorts of related but unwanted molecules.
Right. Side products.
Exactly. So you need analytical methods that are super sensitive and reliable, not just to detect impurities, but ideally to figure out what they are, characterize them, and make sure they're controlled below safe limits.
So you know it's there and ensure it's not harmful.
Precisely. This whole rigorous approach to impurity control, it's fundamental to making safe medicines, especially for complex things like peptides.
And I bet you need to write everything down.
Oh, absolutely. Meticulous records. cGMP demands incredibly detailed and accurate documentation. Every step of manufacturing, every test result, anytime something deviates even slightly from the plan, it all has to be recorded.
Why so much paperwork?
It creates a complete audit trail. If there's ever a problem with a batch later, you can trace its entire history. And it's how you prove to regulators that you followed all the rules.
It's like the ultimate lab notebook for the entire factory process.
Kind of, yeah. But legally binding.
Okay. What about the place where they're made? The building and the machines?
Also covered by cGMP. The design, how they're maintained, making sure the equipment is qualified, meaning it does what it's supposed to do reliably.
Okay.
Facilities have to be set up to prevent contamination. Think air handling systems, water purity, how things are cleaned and sterilized. It's all tightly controlled.
And the people running the machines?
Critically important. cGMP requires that everyone involved, from manufacturing operators to quality control analysts, is properly trained and qualified for their specific job.
Makes sense. Minimizes human error.
Exactly. Trained people following validated procedures consistently. That reduces the risk of mistakes that could impact quality.
You also mentioned process validation earlier as part of cGMP. What's that?
Process validation. This links into guidelines like ICH-Q8 on pharmaceutical development. It's essentially about proving, with solid data, that your manufacturing process consistently produces a product meeting its predetermined specifications and quality attributes.
So it's not just testing the final stuff, but proving the process works reliably.
Yes, it's about understanding your process deeply, gathering data throughout development and manufacturing to show it's reliable and under control. You identify the critical factors that could affect quality.
Like temperature or mixing speed.
Exactly. And you show that even if those parameters vary slightly within acceptable limits, the final API quality isn't negatively impacted. You demonstrate robustness.
So you're basically proving to the regulators and ultimately to patients, we know exactly how to make this consistently well.
That's the essence of it. And doing all this, rigorously adhering to every aspect of cGMP, is absolutely essential for clearing the next major hurdle, regulatory validation.
Okay, so getting the green light from agencies like the FDA, how does that work?
Well, agencies like the FDA in the US or the EMA in Europe, they require extensive validation data before a peptide API can be approved for use in a drug that's sold to patients.
What do companies have to submit?
They submit huge dossiers, things like new drug applications, NDAs,
Yeah.
or for generics, abbreviated new drug applications, ANDAs. These contain exhaustive details about the manufacturing process, all the controls, all the testing.
All that cGMP documentation we talked about.
A huge part of it, yes. Demonstrating full cGMP compliance is absolutely fundamental to getting regulatory approval. You have to show the regulators you're not just saying you follow good practices, but you have the hard data and records to back it all up.
Proof is required.
Absolutely. And the scrutiny doesn't stop there. Regulators also require extensive stability studies.
Stability, how long the drug lasts.
Exactly. You need studies based on specific guidelines to determine the shelf life. How long is the API stable? How long is the final drug product stable under specific storage conditions?
So patients know the expiry date is reliable.
Precisely. You need evidence that the product stays within its quality specifications for its entire intended shelf life. And those tests have to be reliable and meaningful, what they call stability indicating methods.
Makes perfect sense. You need confidence the medicine is still good when you use it.
And one more point here, especially relevant for generics. If a company wants to make a generic version of an existing peptide drug,
Like a copy of a brand name one.
essentially yes, they also have to demonstrate bioequivalence.
Bioequivalence, meaning.
Meaning they have to show, usually through studies in humans, that their generic product behaves the same way in the body as the original brand name drug.
Gets absorbed at the same rate, to the same extent.
Exactly. No significant difference in how quickly and how much drug gets into the bloodstream. It's another significant regulatory hurdle to ensure that the generic is truly interchangeable with the innovator product.
Wow. So even making a copy isn't simple.
Not at all for complex molecules like peptides. So yeah, these key challenges really stack up. The inherent difficulty of large-scale peptide synthesis, the absolute necessity of strict cGMP compliance throughout, and then the rigorous demands of regulatory validation. It all underscores just how complex it is to bring these potentially amazing peptide drugs to patients.
It really is eye-opening. You hear new drug approved and you don't think about all that intricate work behind it. Understanding these steps, it definitely gives you a much deeper appreciation for the whole journey.
Yeah. From the lab bench discovery to actually having that medicine available.
It's far more than just the initial science, isn't it? It's this incredible mix of advanced chemistry, really sophisticated engineering, super stringent quality control, and navigating complex regulations.
A carefully orchestrated interplay, absolutely.
So thinking about all this complexity we've discussed, it does make you wonder, right? As we get better at finding new peptide therapies, what's next for making them?
That's the big question, isn't it? What kinds of novel manufacturing technologies or maybe more innovative regulatory approaches might come along?
Things to help overcome some of these hurdles, make it maybe faster, cheaper, or just more efficient.
Exactly. How can we streamline this process to broaden patient access? Because ultimately, that's the goal, getting these vital medicines to the people who need them. It's a field that's constantly evolving.
A fascinating thought to end on. Finding ways to make these complex processes smoother will be key to unlocking the full potential of peptide therapeutics in the future.